Hi, my name is Michaela Suckiel, and I'm going to be talking about the research I've been working on so far this summer, which is about novel chemical probes used to modulate the polyamide metabolism. So to begin, I wanted to discuss what polyamines are, why they're important, why we're studying them in my lab, and why I'm studying them specifically for this project. So polyamines are small aliphatic polycations that play a very crucial role in various key cellular processes. And this includes things like cell growth, cell proliferation, apoptosis, and gene regulation. Aberrant accumulation of polyamines is often associated with various pathological states, and this includes things like cancers and inflammatory diseases. Polyamine homeostasis within the cell is regulated by three proteins: ornithine decarboxylase, which can be referred to as ODC, its antizyme, which is referred to as OAZ, and the antizyme inhibitor protein, which is AZIN. ODC functions as the rate-limiting enzyme by converting ornithine to putrescine. OAZ binds the carboxylic end of ODC and targets it for proteasomal degradation. ODC is also regulated by its non-catalytic homolog AZIN, which forms a tighter complex with OAZ, thereby disrupting the ODC-OAZ interaction. This results in the rescue of ODC activity and prevents its degradation by the proteasome. ODC is overexpressed in various cancers, and more recently, a resurgence of interest in targeting ODC has emerged as a treatment for neuroblastoma as well as esophageal cancers specifically. More importantly, polyamine uptake by immune cells leads to immune suppression by increasing myeloid-derived suppressor cells, which attenuates T cell function in the tumor microenvironment. Targeting polyamine biosynthesis and transporting using combination drugs have shown promising results so far in depriving polyamines in the tumor microenvironment, leading to the activation of tumoricidal T cells. In turn, the current study proposes the development of novel chemical probes that disrupts ODC function, as well as proposes a mechanism of inhibition for ODC in vitro. So, to begin, we wanted to identify a peptide binder to inhibit ODC activity, and by inhibiting ODC activity, we're inhibiting the dimerization of ODC. So, in search of the best peptides sequence, which would disrupt this ODC dimer interface, thereby inhibiting the enzymatic activity, we utilized 4 different types of computer software. The first one was ROSI Pepti derived, which I was able to identify the best peptide sequence based on the structure of ODC. Using the results from ROSI, an additional sequence was derived based on the crystal structure of OAZ. Subsequently, PepiTract utilizes the peptide sequence to dock the peptide onto the ODC monomer, as well as HDock and FroDock. But HDock and FroDock, instead of using just the sequence, they use the protein database files or the PDD files of the peptide sequence, which allows them to dock the secondary structure as well of the peptide onto the ODC monomer. And as a result, we found that for peptide 1, docking results suggest that the OAZ mimetic of peptide 1 can bind in a similar region where the C-terminal alpha helix in OAZ is located. Additionally, FroDock and PepiTract predicted similar peptide orientations that are very similar to the OAZ helix in the crystal structure, whereas HDock predicted a peptide with a slightly different orientation. For peptide 2, docking results suggested that the OAZ mimetic can bind in a similar region where the N-terminal alpha helix in OAZ is located, while FroDock predicted a peptide that was nearly identical to that of the OAZ structure. In addition, for peptide 2, HDock predicted a peptide with a different orientation as well as a different protein-protein interactions. We also wanted to utilize helical stapling, as helical stapling is known to increase the helical content of peptides that would otherwise have a low helical content. The chemistry involved in this process is a ring-closing metastasis using Grubbs catalyst to introduce the staple. In addition, an electrophilic warhead consisting of electrophilic functional groups would be introduced to react with the cysteine residues. This improves the binding of the peptide to the target protein and results in a higher half-life. This is significant because we wanted to utilize proximity-dependent covalent ligation here, which relies on the reactivity of nucleophilic amino acids such as the cysteines to covalently attach an interacting molecule via electrophilic functionalities. When a molecule or the ligand and a protein, the receptor, is within close proximity, an irreversible thioester linkage is produced, which increases the potency of the ligand. In addition to that, we do have some preliminary experiments which were able to verify the enzymatic activity of ODC with. So to begin, we expressed ODC in the BL21DE3 cell line, and we're able to purify the protein using a nickel affinity column followed by anionic exchange. And then the protein purification was confirmed using SCS page gel, as ODC has a molecular weight of 52 kilodaltons. In addition to that, we also did a fluorescence assay, and for the assay, we wanted to determine the activity of ODC, and we were able to do so using DSMI and CB6. When ODC is converted to putrescine and DSMI compete to bind with CB6, and since putrescine has a higher affinity for CB6 than DSMI does, if the enzyme is active, there should be a decrease in fluorescence, and upon experimentation we were able to see this decrease in the fluorescence, and we were able to make a Michaelis-Menten graph and calculate the Kcat and the Km values, which were quite close to literature values. For future experimentation, we want all peptides will be synthesized and assayed against ODC. The tandem fluorescent assay system will be used to identify and screen the peptide inhibitors. In addition, peptide inhibitors will also be confirmed using alternate assay systems such as LC-MS. Furthermore, CD spectroscopy will be used to identify and confirm the secondary structure of the identified peptide inhibitors, and proximity-dependent covalent ligation will be confirmed and evidenced via mass spec as well as SCS page gel experimentation.